Glycine might be the simplest of the amino acids structure-wise, but it is far from generic. Instead, I classify it as one of the weirdos because its small size means it can take on really awkward tight angles, and so it can provide flexibleness to regions of proteins, allowing them to be kind of loosey-goosey. So far from the actual generic you might think, with just having a hydrogen as its side chain or its unique part, its R group. Another unique thing about glycine is that it's achiral. This means that it doesn't have right and left hand versions. They look the same, unlike the other amino acids. Additional cool things about glycine include how our body makes it and breaks it and uses it for other things, such as serving as chemical signals and being used to make antioxidants and various things. It also has some fun history, how it gets its name, some other fun history about early protein research. So lots of cool things that we can explore through the lens of glycine. So let's dive in. We'll talk about things like the backbone angles, sterics, dihedral angles, Ramachandran plots, hydrophobicity, size, various ways we can classify amino acids, various topics about amino acids. And so this is the first of the amino acids I'm going to talk about in 20 days of amino acids. And so let's dive in.
Glycine is an amino acid. Amino acids serve as protein building blocks or letters, among other functions. All amino acids share some common parts, so they all have an amino group and a carboxyl group, and those are going to be allowing them to link together into chains called polypeptides that then fold up to form functional proteins. How those proteins fold up is going to be determined in large part by a third part of those amino acids, which is the unique side chain or R group. So there are 20 common genetically encoded amino acids, meaning that the genes for proteins have the instructions for putting in those amino acids. They don't need to be modified after the fact or anything like that. Each of these has a unique side chain, and because these have different properties, they're going to influence how the protein folds up and functions in order to accommodate all of their different interests. So we'll talk more each day about how the different side chains of these amino acids influence their properties and how they're used in and outside of the body. Starting with glycine. So glycine, as I said, is one of these weirdos, and it's the simplest structurally. Its side chain or its R group is only a hydrogen. And so sometimes you might see it said as it doesn't have a side chain or doesn't have an R group because it just has a hydrogen, whereas the others have something substituted out for the hydrogen. And a lot of times in biochemistry, hydrogen is kind of like a space filler because we're dealing with organic compounds. So things that are based on a carbohydrate, like a hydrocarbon skeleton. So basically carbons and hydrogens. And hydrogens are kind of like space fillers. And then you can swap out other things. But in the case of glycine, you just have a hydrogen here. And so this is going to make it the smallest amino acid. In numbers term, it's around 75 grams per mole for its molecular weight. As I mentioned, amino acids link together to form chains called polypeptides, which then fold up to form functional proteins. And the instructions for getting them linked together, what order to link them in, are written in the messenger RNA, which is an RNA copy of the DNA gene. And then in the process of translation, what happens is that the ribosome, this protein making complex, is going to travel along that messenger RNA, and tRNAs are going to bring it to the corresponding amino acid. So we're dealing with a codon and an anticodon. So a codon is a three-letter kind of word in the messenger RNA. And then the tRNA is going to have a complementary anticodon, so kind of like a matching sequence, and then it's going to bring in the corresponding amino acid. And so each of these different amino acids has at least one different codon that tells the tRNA to bring that amino acid, or basically the amino acid with the matching tRNA is going to be able to bind, whereas the others will bind strongly, and so the ribosome won't add it to the growing chain. Glycine has the bottom right corner of this codon table cornered. So anything that starts with GG is going to give you a glycine. Um, okay, so what else about glycine? The small size isn't just in terms of like, oh yeah, it's going to make your protein a little lighter if you were to only have glycine. That's not what really we care about when we're talking about size. Instead, we care about how it affects the sterics of the protein of the bond. So when we talk about sterics, we're talking about orientation in space. Now, proteins have the reason why proteins aren't just like all loosey goosey in part, even if you had an all glycine protein, it wouldn't just be like total spaghetti. This is because the backbone of proteins is going to be restricted. So when those amino acids join together to form proteins, they do so by making what we call peptide bonds. So remember, we have the amino group and the carboxyl group. And these are going to basically combine with the loss of water to form a peptide bond. We call this an amide bond, and in the case of proteins, we call it a peptide bond. This is an amide bond. I should also note that although you lose the equivalent of water, when this is actually happening in the body, it's not happening by a simple condensation like this. It's actually happening kind of stepwise, but you lose the equivalent of a water molecule. And you get this, what's special about this peptide bond, about this amide bond, is it has resonance. It has this partial double bonded character. So basically, each of these different circles represents an atom, just one of the basic building blocks of an element. So an oxygen, a carbon, a hydrogen, a nitrogen, these are all different atoms. And they're joint, they have smaller parts called subatomic particles. These include negatively charged electrons, positively charged protons, and neutral neutrons. And the positively charged protons held together by those neutral neutrons hang out in this dense central nucleus. And then this cloud of negatively charged electrons whizzes around it. Atoms can actually kind of merge parts of their electron clouds and share pairs of electrons in order to form bonds. So when you see one of these lines, it represents a bond. And if you see two lines, that represents a double bond. In a single bond, you're only sharing a pair of electrons, whereas in a double bond, you're sharing two pairs. What can happen in something called resonance or conjugation, resonance stabilization. Basically, what happens is you can have these electrons shared by more than just the two atoms. So in the case of a peptide bond, what you actually have is you have this resonance between the nitrogen, the carbon, and the oxygen, where the electrons are kind of getting shared with all three of them. And so it's kind of like you could draw it with having double bond to the oxygen or double bond to the nitrogen. But the reality is instead of going back and forth between these, it's kind of something in the middle. So you have this partial double bonded character with both of these. Why does that matter? Well, you can't turn easily around a double bond. So you can turn around single bonds, you can rotate around them, but when you have a double bond, it's like stuck. It's based on how like the orientation of various orbitals and stuff, which I'm not going to get into. But basically, a double bond you can't rotate around, a single bond you can rotate around. And because you have this partial double bonded character, you're not going to be able to rotate around this. As a result, you end up with this kind of plane where you can rotate freely around here, you can rotate freely around here, but you can't rotate around here. And so if you keep linking up amino acids, what's going to happen is that you get this chain of planes. And we can describe these angles, these psi angles and these phi angles. It can help if you have models, you can kind of make long chains and kind of play around with interpreting the psi and the phi angles and see how that turns out and where you can and can't rotate. But the bottom line is that you have those kind of two main angles that you can rotate around, and how far you can turn is going to depend in part on this R group. So if you have something really small like glycine, you're going to be able to rotate pretty freely. Whereas if you have something big and bulky, well, now even if you could even though you can rotate freely around these single bonds, you're going to get stuck because there's steric hindrance. So basically, remember, sterics is kind of like the orientation in space and the arrangement in space. And hindrance, well, it's like you can't go there. It's kind of like atoms need their elbow room. Two things can't occupy the same space. So if you have something big and bulky, it's going to take up more space, and so it's going to not be able to get as close to other things, and so it's going to have restricted movement. Now, how we represent this restricted movement, we can kind of draw these graphs, what we call these Ramachandran charts. These are going to show you the backbone dihedral angles. So when we talk about these two angles, these are examples of what we call dihedral angles. And we kind of have these angles between these different like planes and stuff. And we can draw the side, we can graph out the side versus unified angles and then show where different amino acids are found. You'll see that in the case of most proteins, you see a characteristic distribution of where these different, where amino acids will be found, like what angles will take on. So often what you're seeing when you're seeing a Ramachandran plot is something that's been, so you have a structure that's been generated by a method like cryo-electron microscopy or X-ray crystallography. I talk about these more in other posts, but they're basic ways where we can get information about the structures of proteins. So we can figure out like, okay, well, what regions are going to be, where are these different atoms located in 3D space? And you'll often see these characteristic structures, things like these beta strands, which can form into these parallel or interparallel beta plated sheets, as well as these alpha helices, these short helices. And you get these characteristic structures because the amino acids tend to take on distinctive side and phi angles that allow them to form favorable interactions with other amino acids. Because you have that distinct spacing of the amino acids, you have that same backbone throughout. And because the amino acids tend to like to take on specific angles, you end up with these specific structures where these amino acids are able to form these favorable interactions, these sort of hydrogen bondings, which are these partial charge attractions between the oxygens and the nitrogens, and between the oxygens and the hydrogens of the backbone of the protein. And so these basically, because you have that ordered arrangement, you have those favorable angles, and you have the ability to form these hydrogen bonds, you end up with these characteristic structures, and these characteristic structures are going to have characteristic side phi angles. And so if you solve the structure of a protein, you figure out what it looks like, and then you take all those different atoms and you plot the angles that they're taking, you get a characteristic distribution. Well, you'll see that there's going to be a high number of atoms that are taking in one position, which is favorable for a beta strand. A lot of them are also taking another position, which is favorable for alpha helices. Then you have some other things like left-handed alpha helices. So if you just took like a generic amino acid, or if you just look at a generic protein, you'd see a distribution kind of like this, where the dark is going to be like generous, like allowed, the core, the medium darkness is going to be like, okay, this is allowed, but it's not like totally favorable. And then the light is going to be generously allowed. When you look at something like glycine, if you look at the regions where it's like allowed or not allowed, you see it looks a lot different. And this is because glycine can take on those weird angles because it has that really small side chain. It has that high amount of flexibility. We'll see that on the other end of the spectrum, we're going to look at later proline, which basically has this really weird side chain where it kind of loops around itself, and it's really restricted in terms of its movement. And so you see this characteristic distribution that's a lot different than the others, but in this case, it's a lot tighter, whereas with glycine, you have a lot more places that it can take up. When people are solving proteins structures, they often plot out your Ramachandran plot and see, okay, well, do the points on my protein, do they fall outside of these kind of accepted regions or non-accepted regions? And that could indicate that you might have modeled parts of the structure incorrectly, because basically with these methods, you're getting the information, but then you have to build the model based on the information. And if your model has it so that the amino acids are taking on really weird positions that are outside of the allowed regions, well, then that's going to be a problem, as that can indicate that there's a problem with the region of a protein that you need to then go investigate further. So glycine and proline are these two kind of like structural weirdos, but they actually work together in forming a really important protein in our body, collagen. So collagen forms this really weird structure called this like triple helix. And basically you have these repeating sequences where you have glycine and proline, and it's a modified proline. But basically, because glycine is so flexible, it enables these prolines to form these chains with glycine, helping kind of give it so that these can form. And this triple helix gives collagen its strength, so we can have strong skin and hair and all of that various stuff. And then if you don't, this basically the modification of proline, it requires vitamin C, and so this is the reason why you have problems if you have scurvy, if you don't have enough vitamin C. You can't make this modification of the proline, and so you can't make strong collagen. But anyway, okay. What else is cool about glycine? As I mentioned before, it's achiral. And so chirality is another one of those words that we can talk about when we're dealing with sterics and stereochemistry, so the orientation in space. All the amino acids except for glycine are chiral. When we talk about chiral, this means that they have non-superimposable mirror images. So this is like your left hand and your right hand. Their mirror images are the same, but if you try to overlap them, they're not the same. And so basically, with most amino acids, you have an L form and a D form, where L and D are kind of like right and left hands. And all of the amino acids your body uses are going to be in the L form. And this is important because all of the enzymes, so all of the reaction helpers, basically mostly proteins that are going to bind to molecules and make them into things like the ribosome is going to help piece together the amino acids. If you were to have the amino acids, well, it wouldn't fit in the pockets of the ribosome. And so different molecules, your body, all the enzymes that deal with working with amino acids are going to be dealing with working with the L forms. But in the case of glycine, we don't have that issue of chirality because you don't have any like chiral centers. Basically, if you were to flip it over, it would look exactly the same, and you could overlay them. So glycine is going to be what we call achiral, whereas the others are chiral. Of hydropathy, so basically, does the amino acid play nice with water? If something is hydrophobic, what's going to happen is that water is going to exclude it. So water is basically, because when we talked about sharing electrons to form bonds, they don't always share fairly. And so what happens in the case of a polar molecule like water is that some of the atoms are going to hog the shared electrons. And remember, those electrons are negatively charged. Oxygen is going to hog the electrons, making it partially negatively charged. And this is going to draw the electrons away from the hydrogens, leaving them partially positively charged, because remember, they have those protons, they don't have the electrons to cancel them out. And so now they're partly positively charged. This is going to make it so that you have this separation of charges. And therefore, the negative part of one water molecule is going to be attracted to the positive part of another water molecule, et cetera, et cetera. And so water is going to form these networks, these kind of cliques. And in order to let something into their network, that thing has to have something that the water likes. So typically, this would be a charged or partial charged thing that could kind of compensate for the loss of a bond to water, and maybe even be better. But if you put in something that is non-polar, so it doesn't have any of that like separation of charge, it's just kind of blah, well, then the water is not going to want to let it into its network. The water is going to exclude it. We call these molecules hydrophobic. So like water fearing, but they're not really feared. The water just doesn't like them. And so the water is kind of going to minimize its contact with them, pushing them into the center of the protein. And this force is actually going to be driving this hydrophobic effect is going to be a main driving force of protein folding. And so we'll see throughout these days that we're going to be able to characterize different amino acids based on how hydrophobic they are. So do they like being found in the center of a protein, or how hydrophilic they are? Are you likely to find them out on the surface of the protein? In the case of glycine, it's kind of in the middle. So it can be found a lot of different places in proteins. But because of that flexibility issue, you're not likely to find it actually in those helices or the sheets. Instead, you're likely to find it in these loopy regions connecting them. And so you see there's some sorts of like sharp turns of things that glycine is really great in order to help the protein kind of link up those more structured regions. And so we'll talk more about this later, but these strands and these helices, these are what we call the secondary structure. And they involve regions, they involve interactions between the backbone of the protein. Okay, so that was the sterics and size and everything, but there's a lot more going on with glycine that we can talk about. And so where does it come from in the body? Basically, glycine is what we call a non-essential amino acid. Now, this doesn't mean that we don't need it. We do need it. It just means that our body can make it from other things. And so in the case of glycine, it can be made from serine. So serine is another amino acid that we'll look at later. And if we compare serine and glycine, we can see serine has this additional methylene group and a hydroxyl group. So it has this CH2OH. We need to get rid of those if we want to make glycine. We need to get rid of the hydroxy group and this methylene group. And so what we can do is we can pass the oxygen and the hydroxyl off onto a proton to make water, and we can pass this methylene group onto tetrahydrofolate in order to form this 510 methylene tetrahydrofolate. So basically, this part is going to become this part. And this can be done with the help of serine hydroxyl methyltransferase, or SHMT. You also see it called glycine hydroxymethyltransferase because it makes this reaction go both ways. So you can go between serine and glycine. So we can get glycine from serine, and therefore glycine is considered non-essential. We can also break glycine down. And so there's this glycine cleavage complex, or this GCC. And basically what it does is it also uses THF, but in this case, it takes this central carbon. It's what we call this alpha carbon, and it's going to take that carbon unit and put it onto the THF instead of taking this carbon unit from serine. It's also going to give you ammonia to get rid of this amino group, as well as carbon dioxide dealing with this carboxyl group. So this will break glycine down. And you can actually do other things with glycine if you either break it down to those building blocks or you just break it down a little. Basically, glycine can be used to form pyruvate. And pyruvate is a molecule that can enter the tricarboxylic acid cycle after it's been converted to acetyl-CoA. And so this tricarboxylic acid cycle is also known as the Krebs cycle or the citric acid cycle. And it can be used to make glucose, so make blood sugar. And so because glycine is able to make pyruvate, which is then able to make acetyl-CoA, which can go into the cycle, basically glycine, it can be used to make glucose, so we call it glucogenic. Okay. Parts of glycine can also be used to make other things. So one of these are the purines. So this would be your adenine. This would be your adenine and your guanine. So two of the bases for DNA and RNA. And so these nucleic acids, they have generic and unique parts as well, and they're unique parts of these nucleobases. And we call these adenine and guanine, we call these purines, and they can get made in part from glycine. Another thing that can be made from glycine is glutathione. So glutathione is made up of these three amino acids, and so we'll see these other ones later, but we have glycine, cysteine, and glutamate, and they kind of link up to form an antioxidant. We'll talk much more about this when we talk about cysteine, but basically cysteine is this amino acid with this sulfhydryl group, and it can get oxidized, where basically it can form these cross links where these two cysteine molecules link together. And this can cause problems if this happens where you don't want it. And glutathione is able to kind of break up those bonds and reduce the protein back to its original state. And so we'll talk much more about that when we talk about cysteine, but glycine is one of the components that's helping make this glutathione to act as an antioxidant. Glycine can also be used to help make porphyrins, which is going to be part of molecules like heme and chlorophyll So in heme, this is going to carry contain iron and help carry help transport oxygen throughout the blood. Basically, these heme groups are going to be held by this protein hemoglobin, and then because these heme groups can help coordinate this oxygen, this hemoglobin is going to be able to transport oxygen throughout the blood. Okay, that was in the body. In the lab, we also use glycine as a buffer. It can be really helpful for this. But one more thing about in the body is it can also serve as like a neurotransmitter, so kind of like a brain signaling molecule. In the lab, we sometimes use glycine as a buffer. So basically, pH is a measure of like how acidic or basic or alkaline something is. So we can talk about basic or alkaline. We kind of use those terms interchangeably. And that would be where you have a low concentration of protons. And something that's acidic, you would have a high concentration of protons. Now, what's kind of confusing is that a low pH is going to be higher concentration of protons, more acidic. And a low pH is going to be a lower concentration of a high pH is going to be a lower concentration of protons, more basic or alkaline. And a low pH is going to be a higher concentration of protons, more acidic. And the reason that this is because it's this inverse log thing. So much more on pH and other posts. But basically, it's a measure of how many freely floating protons there are. And various atoms can give and take protons. They can serve as an acid, which is something that donates a proton, or a base, something that takes a proton. And based on how many acids and bases you are, you're going to have a certain amount of free protons floating around. And that would be a measure of the pH. Now, all amino acids are able to act as acids and bases because that amino group and that carboxyl group can serve as acids and bases. And so acids and bases are two sides of the same coin. So if something acts as a base, it becomes its conjugate acid, and it acts as an acid, it becomes its conjugate base. So you can both give and take. Because once you've given, well, now you can take back. What happens is this amino group and this carboxyl group, they can give and take protons. And so what you get this situation where you're going to have this amino group that we often draw it in this state where it's going to be, where it has this like no charge. This is not like a realistic thing. This is just how it's often drawn. Instead, what's really happening in what's the form that dominates in your body is going to be this Witter ionic form. It says Witter ionic means you have like a positive charge and a negative charge that are separated. And you have fully positive and fully negative parts, but neutral overall. And in the case of an amino acid, this amino group is typically going to be protonated. So it's going to be in its positively charged state. And this carboxylic acid group is going to be in its negatively charged state. So this is a carboxylic acid. It's able to act as an acid, able to donate a proton because it has this proton here. When this acts as an acid, it gives up that proton, and this becomes this carboxylate group, this carboxylate anion. So if you see the terms carboxylic acid, that refers to it having this hydrogen, and carboxylate, it doesn't have this. Although typically we often use these terms interchangeably. And again, although you might see it drawn like this is not physiological, because what happens is that there are characteristic affinities for having a proton or not having a proton. And so depending on how many protons are around, an atom may give or may take. And much more on this in other posts, but we can talk about this on this measure called the pKa. And so that's telling you the pH at which half of the molecules will have given and half of them will have taken. So if you look at this nitrogen group, it has a pKa of around 9 to 10. This means that above a pH of 10, so remember the higher in pH you go, the fewer the protons. The fewer the protons, the less likely something is to be protonated. And so at a really high, you have to get at a really high pH in order for this to deprotonate, in order to get it in that form. In the case of the carboxylic acid group, this is a really low pH, so it's about 2. So anywhere above 2, there's going to be deprotonated. And anywhere below 2, now you have enough protons where it's more likely to have one. And so you can see that this nitrogen wants this proton a lot more than this oxygen wants the proton. And so if you were to go higher in the pH, well, now this is going to be deprotonated, but so is this. This was deprotonated a long time ago. And if you were to go lower, well, then they're both going to be protonated. And you're never really going to see this anionic form. That's just a little advanced because you often see it drawn that way. And sometimes how you can draw it that way because it's kind of convention. But you have these two pKa's, these two different pHs at which these different groups will give or take a proton on average. And what happens is if you average these out, well, now you get something called the pI. So it's basically telling you the pH at which this molecule is going to be neutral overall. This pKa, so you can talk about pKa1 and pKa2 because you have those two groups, and they're going to be slightly different for the different amino acids because it's going to depend on the different R groups. And remember, this is talking about free-floating amino acids because in the case of a peptide, you're only going to have the ends have these free groups. But in the case of the free-floating amino acids, now you have these two groups, and we can talk about having a pi. So for glycine, the pKa1, so for carboxylic acid, is going to be around 2.34, and the pKa2 is going to be around 9.6. And if you average those, you get 5.97. So this is going to give you a pi of around 6. Okay, so why does that matter? Basically, if you were to put a bunch of glycine into a solution, it would make it so that it would adjust the pH to be about 6 and would keep the pH about 6. You have those groups that can give and take protons, and therefore you're going to be able to keep this kind of buffer things if protons are added or taken away, keep things stable at around a pH of 6. And this turns out to be useful for making things like trisglycine buffers for gels. It's just for like SDS page. If you're running a trisglycine gel, this would use glycine as one of the buffering agents along with tris, which is another buffer. It's also used as a buffer in toiletries and cosmetics. So about 15,000 tons of it are made commercially each year. Another fun fact about it is it's actually been found in outer space. And so NASA found it in samples taken from comets, and it was the first amino acid to be found in space. So, or found in a comet. So pretty cool stuff. And that was like 2009, and I'll link to it. Okay, just some other fun history. So glycine, if we look at the early history of protein chemistry, basically originally proteins, people didn't really know what proteins were, and they kind of described them as like three main categories. So you had albumin, fibrin, and gelatin. So that's how they were described in early days by Antoine Fourcret. The first amino acids were actually isolated in 1809, but they weren't really sure that it was actually part of a protein. Then what happened in 1820 was the scientist Henry Bracknott. He was the first to isolate amino acids by acid hydrolysis. So basically, he broke apart those different amino acids using acids to take that peptide, take that protein, and break it up into these individual amino acids, and then try to identify those amino acids. And when he did this, he actually discovered glycine. And so glycine was, depending where you look, glycine or leucine was the first to be discovered by acid hydrolysis. And in this case, Henry Bracknott, he was actually looking to make sugar. So he knew that if you were to treat like wood or straw with acid, you get sugar. And he wanted to see if the same thing would happen with animal products. And so he took gelatin and he boiled it with sulfuric acid and neutralized that acid. And then he left it to crystallize. And it crystallized, and he tasted it, which you should never do with things that you do in the lab. But he tasted it, and it tasted sweet, and so he thought it was sugar. But he was actually wrong. He didn't know that, but he named it sucrated gelatin. And then that was translated into German as Leinzucker. And he did some of the characterization of its solubility and things. But remember, he was still thinking it was sugar and didn't know it was a protein part or anything like that. But then later scientists were going to follow up on his work and study it further. So even Roy Horsford, he didn't like that original name because he's like, well, this isn't actually a sugar. And so he suggested the name glycol, which means sweet glue in 1846. And then a couple years later, Berzelius said he didn't like that name and it didn't really match with the other amino acid names. So he suggested the shorter name that stuck, glycine. And so that's how the history of glycine. And then later we'll talk more about some other key milestones in protein chemistry. But remember, at this point, In 1820, when we discovered this, we don't really even know what's part of a protein. We don't really know what proteins are. We don't know what peptide bonds are. We don't know what the structures are. But it was this important early discovery. And so, just to sum it up, glycine is the smallest amino acid, but it's not generic. When we talk about generic, that's going to be more like our next amino acid, alanine, which is totally generic, but we typically use it as kind of like the most generic. It had but with glycine, we just have this hydrogen, so it's going to offer a high amount of flexibility. And so glycine, you'll typically find it in loopy regions of proteins. We also find it in this collagen triple helix. It can allow other areas of proteins to take on more complex forms. It's a chiral, so it's right and left-handed versions look the same. It is going to be non-essential, which means that our bodies can make it ourselves. In this case, our bodies can make it from serine, and we can do other things with it, like make purines and glutathione and porphyrins. So glycine is a really cool amino acid, and it's a really important amino acid, and our bodies use it. We use it in the lab, and I hope that you can now appreciate why it's really fab.